Welcome back to this next video, and in this video we are going to talk about the two-dimensional gel electrophoresis. So the first thing is, what is two-dimensional gel electrophoresis? The two-dimensional gel electrophoresis, also known as the 2DE, this is the two-dimensional electrophoresis. It is also known as the two-dimensional polyacrylamide gel electrophoresis, because in the two-dimensional gel electrophoresis, the gel or the matrix that you are using, that is polyacrylamide. So it is also known as the two-dimensional polyacrylamide gel electrophoresis. So the 2DE or the 2D page, that actually refers to one in the same thing. Now the 2DE or the 2D page, that actually separate the complex mixtures of the samples containing proteins using two different properties of the proteins in this particular mixture. Now the two properties that the two-dimensional gel electrophoresis that are exploiting for separation of the proteins, the first property is known as the isoelectric point, and the technique which utilize the isoelectric point is a source of separation of the protein is known as the isoelectric focusing. So the first dimension of the 2D, that is known as the isoelectric focusing, which actually separate the proteins based on the differences in their isoelectric point. Now the second dimension of the two-dimensional gel electrophoresis, that is the molecular weight, and the technique which utilizes the molecular weight is a property of separation of different proteins from each other, that is known as the sodium dodecyl sulfate polyacrylamide gel electrophoresis, for short, which is known as the SDS-PAGE. So the second dimension of the 2DE is the SDS-PAGE, which actually separates the the different proteins in the mixture based on differences in their molecular weight. So this is one reason why it is called is the two-dimensional gel electrophoresis. The other reason you can call it is the two-dimensional is that, as you will see in a while, that the first dimension of the proteins, when you are separating the protein based on the isoelectric focusing, you are actually separating them in a horizontal manner. The strip that you are using, that is actually placed on a horizontal surface, and the protein, they get separated from each other in a horizontal dimension. When you talk about this SDS-PAGE, this is actually the vertical dimension. So as you are using two dimensions, the horizontal and the vertical, this is another reason why it is called is the 2DE. So it is called as 2DE because of two things. It have got a horizontal and a vertical dimension, and it is separating the protein based on two different properties. Now what you do is that when in the first dimension you separate the proteins on the gel with isoelectric focusing, those particular proteins are then negatively charged by the treatment with the SDS, and the electrophoresis is performed by inserting the gel horizontally into the SDS-PAGE gel. You will see that in a while. Now the power of the 2DE or the 2D page is that it is very unlikely the two molecules, they will be similar in two distant properties, and molecules are more effectively separated in 2D electrophoresis than in one-dimensional electrophoresis. For example, if you are separating the protein only on the basis of the isoelectric point, there can be chances that two proteins, they may be having a very close isoelectric point, and you cannot actually then differentiate between the bands of those two proteins. Alternatively, you can also have certain proteins which are having very close molecular weight. So if you are separating them only on the SDS-PAGE, their molecular weight, because of the close values of their molecular weight, you may not be able to differentiate between the bands of the two protein. But if you are applying two dimensions on a single sample, so for example, we say that the two proteins, they have got the same isoelectric point, but they are different in their molecular weight, they will get separated in the 2D gel electrophoresis. Or if they have got the same molecular weight or similar molecular weight, but if they have got differences in the isoelectric point, again they will get separated from each other. So this is the power of the 2DE, that it is very unlikely that two molecules or two proteins, they would be similar in both of the properties. Therefore it is very effective that the 2D electrophoresis, that is very effectively separating the proteins from each other. If you talk about the history, it was described, the 2D that was described back in 1975 by O'Farrell, but its applicability and adoptability were enhanced because of the introduction of the immobilized pH gradient strips, which are used in the isoelectric focusing, and we will see that in a while. Now these immobilized pH gradient strips, they have very good reproducible results, and handling become very easy. Another important advance you can see that is made in the 2DE is that initially the protein, they were visualized by using the radioisotopes of the phosphorus and the sulfur, and is the radioactivity that can be damaging to the individual dealing with these samples. So currently these radioactive isotope that has been replaced by more sensitive techniques of detection or visualization, such as the cypro-ruby. So the cypro-ruby, we will discuss that in the staining videos, that how you can actually stain your proteins after they have been separated in the 2D page by the cypro-ruby. So this cypro-ruby is actually a fluorescent probe which can actually help you in visualization of the proteins. Advances were made in different stages of the 2D pH technique, now which are capable of separating up to 10,000 proteins in a single gel, which have a dimension of 20 into 20 centimeters. So in the 20 to 20 centimeter gel, you can actually separate about 10,000 proteins, and that is a very great achievement or a very great advancement in the 2D pH. Now a 2D pH image gel is captured, and the image analysis is done to find out the number of protein expressed in a particular sample or in a particular tissue. Now another way you can actually visualize the protein is by using different kind of the stains, and we will discuss the stain as well in a separate video. But if the protein amount is around 10 nanogram, the most suitable stain that is used, that is known as the Comasier dye that is used. But if the protein amount is low, like if it is around 0.5 nanogram, the options, the dye that are available, you can actually use the silver or the fluorescent coated protein stains that can be used for the detection. Now this image is actually an overview of the whole process. What you do is that you horizontally separate the proteins by the technique of the isoelectric focusing, which separate the protein based on their isoelectric point, and when they get separated by the isoelectric point, this is then inserted into the two-dimensional gel, which is actually in the SDS-PAGE, not the two-dimensional gel, the second dimension of the 2D page, which is the SDS-PAGE. And as you can see over here the separation that is vertical, here the separation is horizontal, here the separation that is vertical, and the protein, they can actually get separated from each other by utilizing the 2D gel, the isoelectric focusing, and then the SDS-PAGE. When you talk about the 2D, whether you talk about the isoelectric focusing or you talk about the SDS-PAGE, in both of the cases, the matrix or the polymer that is used, that is the polyacrylamide. So you should have a firm understanding what is polyacrylamide and how the polyacrylamide is made. Now this polyacrylamide is very ideal for the protein separation because it is chemically inert. One of the greatest properties of the polyacrylamide gel, that they are chemically inert, so they are not going to interact in any way with your proteins. They are electrically neutral, so they are not affecting the movement of the proteins when you apply the electric charges. They are hydrophilic, they are transparent for optical detection at wavelengths greater than 250 nanometer, and it has a very low affinity for the common protein stains that we just discussed. It has a very low affinity for the Comasier stain, it has a very low affinity for the silver stains. So because of these properties, this polyacrylamide, it is very ideal for the separation of the proteins. Now when you talk about the process of polymerization of polyacrylamide gel, you actually need four things. One, you need the monomers of the acrylamide, you need a cross-linker, which is the N,N-methylene bisacrylamide. So this bisacrylamide that is actually working as a cross-linker, you will see that. You need ammonium persulfate or a riboflavin, and the APS or the riboflavin that actually acts as a source of free radicals that react with the vinyl group in the acrylamide and bisacrylamide, thereby activating them. Initially, these acrylamide monomers and these methylene bisacrylamide monomers, they are inert, they are not very reactive, but when you add the APS or the riboflavin and when they produce the free radicals, these free radicals are actually going to make the acrylamide and the methylene bisacrylamide active. So you can actually have the polymers of the acrylamide from the acrylamide monomers, and when the monomers of the acrylamide they polymerize, you are getting the acrylamide polymers. When you get these acrylamide polymers, this N-methylene bisacrylamide is actually cross-linking those polymers of the acrylamide, thereby giving you a gel structure. Now the catalyst that is used in this whole process, that is known as the tetramethylene diamine for short, which is known as the TIMID, and it acts as a catalyst in the polymerization of the polyacrylamide gel. If you see over here, these are actually the vinyl groups over here. In the this is the vinyl group in the methylene bisacrylamide. These are the vinyl groups in the acrylamide monomers. So in the presence of the free radicals produced by the APS or the riboflavin, these vinyl groups they actually get activated. You are actually getting the polymers of the acrylamide, as you can see over here. So for example, if this is the polymeric structure of the acrylamide, this bisacrylamide is actually cross-linking them. It is actually cross-linking the polymers. If this is one polymer, this is another polymer, this is another polymer, this is another polymer. So the methylene bisacrylamide is actually cross-linking all of these acrylamide polymers, thereby making the polyacrylamide. Now this polymerization speed, that depends on various factors. It can depend on the monomer and the catalyst concentration. It depends on the temperature and the purity of the reagents. And the polymerization speed, that depends on various factors, and these factors must be carefully controlled, since this process generates heat and polymerization that if too rapid, it may lead to non-uniform pore structure. So if you are not able to control these factors, that means that you cannot actually have a control over the pore structure. So the if you are not controlling these things, you in the end you will be getting a polymer with a non-uniform pore structure, which can actually affect the separation of the proteins. Now the pore size, that is actually controlled by the investigators. They actually control the size of the pores in the gel by adjusting the concentration of the acrylamide. So this acrylamide concentration is actually used to control the pore size in the gel. The second important thing is the ratio of the acrylamide to bisacrylamide. That is very important, because the ratio of the acrylamide and the bisacrylamide, as you can see over here, is actually the determining factor for the pore size of the gel of the polyacrylamide. So the first dimension, as I've told you, in the polyacrylamide gel, that is the isoelectric focusing. Now the isoelectric focusing, which is also known as the electrophoresing, that is actually a technique which separates different molecules by differences in their isoelectric point. So as we are focusing on proteins over here, so the isoelectric focusing is actually separating different proteins by differences in their isoelectric point. So the first thing is, what is an isoelectric point? As you can see over here, we are talking about an electric point. But this term iso, it actually refers to the same. So that means you are having same electric point. So the isoelectric point, which for short is known as the PI or the PHI or the IEP, it is actually the pH at which a molecule carries no electrical charge, or it is electrically neutral. It do have a charge, but it is electrically neutral. Now this IEF or electrophoresing, it is actually a type of the electrophoresis that is usually performed on proteins in the gel. They take advantage of the fact that overall charge on the molecule of interest, that depends on the pH of its surrounding. Now the IEF, that actually involves the use of immobilized pH gradient strips. In the process of the isoelectric focusing, you are using these IPG strips, and I'll show you how you can actually use these IPG strips so that you can separate the proteins based on their isoelectric point. Now these IPG strips, they are actually the acrylamide gel, they are the polyacrylamide gel, which are copolymerized with a pH gradient. That means you will be having a polyacrylamide gel, but the pH of the polyacrylamide gel at different spots, they will be different from each other, so you will be having a gradient of pH on this polyacrylamide gel. So this pH gradient, now this pH gradient, it actually results in completely stable pH gradients except for the most alkaline pH values. Now how this immobilized pH gradient that is made? So the immobilized pH gradient is obtained by a continuous change in the ratio of the immobilins, and this immobilins is actually a weak acid or weak base. So we call this is the immobilized pH gradient because it is you can say copolymerized with the acrylamide. So this pH gradient, as it is not moving, it is immobilized, therefore we call them is the immobilized pH gradient. Now when you talk about the proteins, these proteins, they are actually made up of the amino acids. So this is actually the charge on the amino acid that determine the overall charge of the protein. So say for example, if this is a generic structure of an amino acid, so this amino acid actually can have 3 charge state. At the neutral, which is known as the isoelectric point, this iso mean equal. So at the isoelectric point, say for example, if this is the isoelectric point of a particular amino acid, so at the isoelectric point, as you can see over here, it have got a negative charge, it have got 1 positive charge. So 1 positive and 1 negative, that means that the molecule is still charged, but as the positive and negative they are equal, that means they will be carrying no net charge, they will be electrically neutral. When you take this amino acid or this particular protein from its isoelectric point towards a pH which is below its isoelectric point, it will actually be having a positive charge, because if you are moving towards the acidic pH, that means that the concentration of the hydrogen ion that will be increasing, and this amino group over here, that is actually an acceptor of the hydrogen ion. So if it is accepting a hydrogen ion, that means it will be carrying a positive charge. If you are moving an amino acid or a protein towards a pH that is above its isoelectric point, as you can see over here, the concentration of the hydroxyl ion that will be increasing. The amino group will not be available with any proton to get accepted and hence carry a positive charge. So on the proteins there will be a negative charge, or on the amino acid there will be a negative charge if it moves towards a pH that is above its isoelectric point. But at the isoelectric point, it is actually electrically neutral, and when that is electrically neutral, that means it will not be carrying any charge. When you apply the mixture of the protein to the IPG strips, as I will show you that in the slides to come, so when you apply that, the protein that will start moving in the pH gradient, because initially that will be charged, they will be positively charged or they will be negative charged. So if they are having a positive charge, they will start moving towards the negative end. If they have got a positive charge, if they have got a negative charge, they will start moving towards the negative end. So when they start moving through the pH gradient, the protein all are charged that will change until the protein reaches the pH region that corresponds to its pi. And when the protein reaches its isoelectric point, at that particular point it will be electrically neutral, and if that is electrically neutral, that means it will not be moving in the electrophoresis, because in the electrophoresis only the charged molecule they can move. So when they reach the isoelectric point, there will be no net charge on them. That means they will be electrically neutral, so they will stop moving in the IPG strips, because at this point it will have no net charge, so the migration it ceases. Now as a result, as the different proteins have got different isoelectric point, so as a result the protein they become focused into sharp stationary bands with each protein position at pH of its pi. Now this technique is capable of extremely high resolution, which protein differing by a single charge being fractionated into separate bands. As you can see over here, that if this is the pH gradient and if your mixture have got these two proteins, so they will start moving. But when they reach their isoelectric point, say for example if the isoelectric point of this particular protein is over here, so when it reaches over here it will stop moving because it will be electrically neutral. Similarly, when this protein, if this is the isoelectric point of this particular protein, it will stop moving over here, it will stop movement because it will be electrically charged. Now when you talk about the polyacrylamide gel that you use in the IPG strips, they are actually made with large pores, usually to eliminate any sieving effect, because you are separating the protein based on their isoelectric point, so you will be making the pores very large. That means you will be using a very low concentration of the acrylamide and the bisacrylamide, so you will be having large pores so that the movement of the proteins they do not get impeded by the smaller pore size of the gel. So what you do is that in the isoelectric focusing in the IPG strips, you are using a low concentration of the acrylamide and the bisacrylamide, so you will be having large pores. Now the isoelectric focusing can resolve protein that differs in isoelectric point value by as little as 0.01. So this is actually just a single example, the way the two proteins they stop moving when they reach their isoelectric point. Now this is the IPG strips, and if you can see over here, this IPG strip, two important things are mentioned on each and every IPG strip. One, it is actually showing you the terminals, that this is the positive terminal, so the other end that will be the negative terminal. So it actually tells you about the position of the electrodes where you have to keep this. Secondly, it actually tell you about the pH gradient that is present on this particular IPG strips. And if you can see over here, it is from 4 to 7. That means that this polyacrylamide gel or this IPG strips, it actually have a pH gradient which starts from 4 and it moves up to 7. That means you can only separate those particular proteins on this IPG strips whose isoelectric point it falls in this particular pH. If they are above or below, that means you have to use another kind of the of the IPG strips. So what you do is, if I give you a trip how you are actually performing the the isoelectric focusing. So the first thing is that you have to remove the protective cap and you have to apply the rehydration solution to the strips. Usually the IPG strips, when a cap is over here, they're actually dehydrated. So before using them, you have to apply the rehydration solution, which usually come with the kit of the isoelectric focusing. The third thing is, what you have to do is you have this particular tray over here, and in this particular tray you have to wet the entire length of the IPG strips in the rehydration solution in this particular strip holder. But you have to make it sure that the gel side that is down and the gel side is actually interacting with the rehydration buffer. Then what you do is, when you go for the rehydration, you have to gently lay the entire IPG strip in the strip holder so that the rehydration buffer that uniformly interacts with all of the IPG strips. Another thing, then next what you do is that when you have gone for the rehydration of the IPG strips, you have to apply your protein sample, and you have to spread your protein sample on the entire length of the IPG strips, because when you when you apply the protein whole on the IPG strips, and when you apply the electrical charge, the protein will start moving towards their electrode. Say for example, if you apply the protein sample entirely on this surface, and if this is the positive end, so if a protein is negatively charged over here, it will start moving towards the positive end. And if it is positively charged, and if that protein is present over here, that protein will start moving towards the negative end until their isoelectric point, that is reached, and then they then they cease to move in the IPG strips. And the last step is that you have to place the cover on the strip holder, so that to eliminate the the environmental variations. This is actually the tray or the strip holder when you are using the multiple IPG strips. But one of the important thing to keep in mind is that you have to prevent the formation of the air bubbles, because the formation of the air bubbles, that can actually create problems resulting in vertical streaking. And when the bubbles they are there, they are actually affecting the separation of the proteins in the IPG strips. So if you like the video, please subscribe to my channel, hit the like button, and share it with your friends. And in the next video, I'll be talking about the second dimension of the 2D page, which is the SDS-PAGE.